Hi, thanks for joining us. I'm Jerry Giesbrecht, and along with my postdoctoral fellow Marcel van de Wouw, we will tell you about our work on associations between the gut microbiota and internalizing behavior in preschool children. I want to define what I mean by internalizing, because it's not a term that we use in everyday conversation. In young children, we often use the term internalizing to refer to behaviors that look like anxiety, depression, somatic complaints, or withdrawal. These behaviors are the early indicators of what may later become a mental health problem. We are interested in mental health problems in children because they are quite common, because they cause a lot of suffering, and because they're treatable. If we can intervene early enough, we can increase the developmental potential of children, and we can reduce the huge annual costs associated with mental illness, which now top $51 billion per year in Canada. Recent discoveries about the connections between the gut and the brain have opened up new possibilities for mental health research. We know that there is a continuous back and forth communication between the gut and the brain. The gut produces many metabolites and hormones that can influence brain function and behavior. And of course, our psychological experience can change the ways that our gut works. I'm sure we've all experienced butterflies in our stomach prior to an important event. And this just illustrates how the gut is closely connected to brain and behavior. One of the questions that we've been thinking about a lot recently is how the microbiota that live in our guts, and there's a lot of them, might be involved in development. We know that development is influenced by the experiences and environments that we live in. Development is plastic or moldable by those experiences and environments. During different phases of development, some systems are more open to input than others. This is sometimes referred to as critical or sensitive windows of development. And it's not just that development is more open to input during that window. It actually needs or expects it in order for normal or typical development to occur. So it's not just what actually happens that can influence development. Sometimes it's also what doesn't happen. These are general developmental principles. So if we start with the principle of expected inputs and the fact that humans have evolved in the presence of microbes, then the question that we ask is whether the brain expects microbial inputs at certain times of development. It's a lot easier to address some of these developmental questions using animal models, and a very useful model is the germ-free mouse. These mice are born and raised without any microbes, and this allows us to study the ways that microbes might influence development. These mice are pretty messed up in many ways, so the biggest lesson we can learn is that mice do not develop like mice should develop if they don't have microbes. But the other thing is that there is a critical window in which many of these messed up behaviors can be rescued, and the time frame is very early in life. So the bottom line here is that microbes are important for normal development, and specifically the microbes that we encounter in early life have a big influence on development. Microbes can still have a big influence in later life too. For example, even healthy adults who consume some kinds of pre- and probiotics tend to feel better psychologically, and we see changes in brain function that are also positive. So we know that even among adults, microbes can improve mental well-being. But there are basically no studies in children, so we know very little about the ways that microbes might affect brain and behavior in children. The figure on the right shows the relative amount of gut microbiome data that is currently available in the literature, separated out by age group. We see in the yellow and black, very young infants and older adults have a good amount of focus, and then the lion's share goes to adults. The two blue spaces are tiny in comparison, representing children and adolescents. From the few studies that are available, we know that specific microbes are associated with infant temperament, and as we see in many other exposures, the specific effects depend on sex. One previous study among toddlers found that Previtola was associated with internalizing problems. So from the few studies that were done in children and the studies in adults and animals, we see potential connections between the gut microbiome and mental health, or early indicators of mental health. I'll turn the rest of the talk over to Dr. Van de Wouw.
Thank you, Dr. Giesbrecht. What's important when studying the microbiota is to consider the factors that influence which microbes live and on our body. And in particular, diet seems to be very important. And it makes sense that what we eat is a major determinant of the composition of our gut microbiota. Because what we eat passes through our gastrointestinal tract and essentially becomes the food of these microbes. Typically, these microbes break down that food and then convert these into compounds that may in turn influence us. I would specifically like to focus on dietary fiber. Now, it's nothing new that eating sufficient dietary fiber is important for health. But what's interesting is that these dietary fibers that our own body can't break down can actually be broken down by our gut microbiota, and they convert them into compounds called short-chain fatty acids. The production of short-chain fatty acids is highly dependent on the gut microbiota, as mice without a gut microbiota, germ-free, have almost no short-chain fatty acids. In addition, taking antibiotics that generally deplete the microbiota also results in drastically reduced levels of short-chain fatty acids. As I mentioned, short-chain fatty acids is also to some extent an energy source, and the total amount of short-chain fatty acids can provide up to 10% of your daily caloric intake. And I'd also want to point out which of the three main short-chain fatty acids are within our system, which are, as you can see at the bottom, acetate, propionate, and butyrate. And it's very interesting to also note that especially butyrate has been implicated in colonic health, where it's one of the major energy sources of cells within the colon. Microbiota-derived short-chain fatty acids have already been associated with the brain and behavior as well. This happens through a variety of different mechanisms, and some of these mechanisms include receptor activation, the promotion of serotonin production and synthesis, epigenetic mechanisms, the vagus nerve, and to some extent, some of these short-chain fatty acids can also directly affect the brain as well. Now we're just going through an example of how specific fibers can increase short-chain fatty acids and how this can affect behavior. So, for instance, in this study, they administered fructooligosaccharides and galacto-oligosaccharides to mice. And not very surprisingly, there was an increase in acetate and propionate levels in the gastrointestinal tract of these mice, as can be seen in figure A and B. In addition, the supplementation of these two particular fibers was able to reduce anxiety-like behavior in these mice, as can be seen in figure A, as well as in figure E. And what's interesting to note is that the combination of these fibers was actually most potent. And something similar was observed when looking at depressive-like behavior in these mice, where the combination of both fibers reduced depressive-like behavior, as can be seen in figure C and D here. And once again, when looking at acute stress responses, these two fibers together were most potent at reducing physiological stress responses as measured by corticosterone levels, as can be seen in figure A and B as well. If we look at human research, we see that these particular short-chain fructooligosaccharides are able to reduce anxiety scores in patients with IBS, so irritable bowel syndrome. So the take-home message is that these particular fibers that are known to increase short-chain fatty acid levels can have some beneficial effects on specific aspects of mood. And in the next slide, we'll talk a bit more about what happens when these short-chain fatty acids are administered directly. Here we see an example of this. I would first like to bring your attention to the red bars. Mice that previously underwent chronic stress show a greater response to future stressors, as can be seen with comparing the red bars in figures A and C. Now, what's interesting is that if mice additionally received short-chain fatty acid supplementation, as can be seen in the green bars, then some of these specific effects are ameliorated by the short-chain fatty acids. And more recently, Something similar has been shown in humans, where we see that short-chain fatty acid supplementation can actually reduce the response to an acute stressor in healthy adults. So this brings us to the hypothesis of this project, where we wanted to investigate whether child behavior was associated with the gut microbiota and these compounds called short-chain fatty acids. Internalizing and externalizing behaviors were assessed from 248 preschool children using questionnaires, and internalizing is defined as a group of negative emotions and behaviors focused inwards, whereas externalizing is defined as a group of negative emotions and behaviors that are acted out. We also collected stool samples where we wondered which particular bacteria are present in the microbiota, and we also quantified short-chain fatty acids in the stool. When we look at the population characteristics of this study, we tend to see that participating children had few socio-demographic risk factors. In addition, mothers had mean scores of perceived stress, anxiety, and depression under clinical cutoff scores, indicating that this was a relatively healthy population. When we analyzed child behavior in relation to their gut microbiota, we observed that children with a less diverse gut microbiota tend to have increased internalizing behaviors. Interestingly enough, this was linked to increased somatic complaints, which are physical symptoms such as pains and aches without any medical explanation. When we then focused on children clinically at risk for internalizing behaviors, we also observed that these children have a less diverse microbiota compared to other children. We then correlated infant behavior with short-chain fatty acids in their stool. We found various short-chain fatty acids to correlate with both internalizing behaviors and somatic complaints. But what I would specifically like to point out here was that acetate and butyrate had a negative association with somatic complaints. We then focused on children clinically at risk for somatic complaints and showed that these have less acetate and butyrate in their stool, as can be seen in the figure below. This is very interesting because in particular butyrate has been associated with gastrointestinal health and complaints. Considering that somatic complaints are so highly linked to gastrointestinal complaints, it led us to the concept of, well, can we increase these short-chain fatty acid levels by, let's say, for instance, dietary fiber supplementation, and in that way reduce abdominal pain, and maybe also reduce anxiety or maybe increase quality of life. And there was another study that was performed a few years ago where the dietary fiber psyllium was supplemented to children with irritable bowel syndrome. And here they showed that this supplementation reduced the number of pain episodes that these children had compared to the placebo. This has led us to the next study that we're currently setting up. In this study, we are planning on supplementing fiber, in this case psyllium, which is similar to the previous study I just mentioned, to children with functional abdominal pain. Functional abdominal pain is to some extent relatively similar to IBS, where the biggest difference is that the pain doesn't occur in combination with any bowel movement or something of the sorts. So this is a slightly different population. And in addition, what we're really interested in this case is also understanding whether this whether psyllium supplementation is able to improve anxiety scores and quality of life scores. In addition, what we're very interested in as well is understanding how psyllium might be able to improve abdominal pain and anxiety and quality of life. And we're hoping on understanding this better by investigating aspects of the microbiota-gut-brain axis that are associated with this signaling between the gut and the brain. And in this case, we're investigating the gut microbiota, the vagus nerve, which is the nerve that runs between the gastrointestinal tract and the brain, as well as brain networks to understand which brain regions are impacted. This brings us to the end of this presentation. I would like to thank you all for your attention throughout the presentation, and in particularly, I would like to thank the organizers of this conference for allowing us to present these data. If there are any questions that you have, please feel free to ask them.